Hello, dear listener. I would like to present to you here the poster Transcriptomic Deconvolution and Machine Learning in Rare Neoplasms for the poster presented at the ECCB 2021. My name is Raik Otto. I'm a bioinformatician at the Humboldt University Berlin. Let us start with the introduction and motivation for this poster. The introduction and motivation for this poster is personalized therapy. What this means is that in the current focus of research stands the idea that individualize the treatments of neoplasms. What this means is you look at the cancer patient, you obtain some biomaterial from the neoplasm, something that informs you about the genotype of the neoplasm, you then sequence this neoplasm, and then you model the neoplasm. You try to understand it, you try to derive information, try to understand the mutation and expression data in a computer with machine learning models. The outcome of that ideally is that you can individualize the treatment, that you can find the optimal drug treatment to fight the cancer, that you can prolong the survival of the people suffering from cancer and reduce monetary costs. Now, the problem, however, associated with that for rare cancers are manifold, and this is what this poster is attacking. This is what we're trying to solve here. In case of rare neoplasms, you very often have very limited training material for the machine learning models, which precludes the individualization of the therapy. Now, what this means is that you have a very reduced incidence rate, and because the incidence rate is low, you have very little neoplasms to sequence, which then means you have very little data to train your machine learning models on, especially if your rare cancer is biologically diverse, where you need a lot of training data. And this is what this poster is addressing with transcriptomic deconvolution, which I will explain in a second. We are trying to ameliorate the training of machine learning models for rare neoplasms. Now, how we do that is we, in terms of concept, are applying two main ideas. The first one is the dimensionality reduction, which tries to project the data onto a hyperspace where you can more easily train your machine learning models, and we try to linearize the decision boundaries, thereby improving the prediction performance of the machine learning model. So dimensionality and reduction and linearization are the two key concepts. After you did that to your data, after you transformed your data, you then go ahead and take this transformed data and train your machine learning models on such a data. You evaluate whether the results are good. And in our particular case, we didn't just stop there. We as well compared the results of such trained machine learning models with the results of the gold standard marker KR67 for grading and the originally substituted data. So the data that was not changed. This is what we did. And the way how we did that is we applied the transcriptomic deconvolution. Now, transcriptomic deconvolution fulfills the purpose of dimensionality reduction and linearization. What transcriptomic deconvolution does is it is trying to reconstruct a given patient-derived bulk RNA sequence neoplasms with relative cell type fractions. It tries to reconstruct the neoplasm such that by assumption it only consists of pre-trained cell type proportions. Now, this reconstruction with cell type proportions as a result, for instance, 80% cell type A and 20% cell type B, it's the vector you get, as well as a reconstruction error associated with that. And this is the step that is performing the dimensionality reduction and linearization, which you then, with such transformed data onto the space of cell type proportion prediction, you then train your machine learning model. I would now like to show you how such an approach, such a framework as we have developed, now performs on the benchmark data. The benchmark data we took, the rare neoplasms we looked at, were the pancreatic neuroendocrine neoplasms from 7 studies with about 330 panels, which, however, were not really comparable. We could only use roughly 80 to 90 of them because the rest was not technologically or biologically homogeneous enough. We trained 2 deconvolution models, of which I will only talk about the second one. The second model consisted of the alpha to delta cells, which is where neuroendocrine neoplasms originate from. So that's absolutely biologically sound. But in addition, we added, for reasons I cannot explain, we added the ductal and acinar cell type proportions, where in theory this particular cancer cannot originate from. Now, we only used expression data, and the deconvolution was possible, which I cannot show you due to time constraints. So it was possible to deconvolve the neoplasms. What you get is, for instance, here aggregated by the gradings G1, G2, G3. You get relative cell type proportions, and as you see, the bottom line here is that G1 roughly resembles the healthy pancreatic endocrine stratification, whereas for G2 and G3 you see an increase of ductal proportion. This means there's a correlation, and this correlation you can learn. Let us quickly look at the hyperplane or hyperspace where we project our data into, and what you see here is the projected data. This is data as the method projects it. What you see is that the net and max subtype, which I cannot explain due to time constraints, and the grading can be linearly separated. It's not perfect for various reasons, which is acceptable, but as you see, you can stratify your data, you can train a model on that. And now that is what we did. We, in the next step, trained a random forest, and we tried to predict the grading and the subtypes. And what we found in the comparative analysis, so we're comparing the gold standard with our model, what we found is sensitivity was very good with 91%. The gold standard model even performed slightly worse. Specificity was very comparable for such a model. So we concluded in that sense that the predictive performance was comparable. And same for the survival time. Here you see two survival curves. Both of them, one of them is the gold standard that clinicians would use. The other one is the ductal cell type proportion. So as you can see, some patient survival time can be predicted as well. Now, I just want to raise your awareness that there's a certain conflict using the ductal cell type proportions, but this type of cancer technically cannot arise there due to, according to the clinicians. We just want to point that out. To conclude, the transcriptomic deconvolution is very useful because you don't require neoplastic data, and the machine learning performance is comparable to the gold standard. Now, what we did not show, but what is a problem, is that the optimal combination of training data and of the convolution algorithm has been found, and that no gold standard for the proportion predictance exists. These are my image credits. Thank you very much.